Hi everyone, welcome to the CCMP seminar series. I would like to begin by acknowledging the Wurundjeri people of the Kulin Nation as the traditional owner of the land on which our Parkville campus stands. I also acknowledge the traditional custodians of various lands on which you all are joining me from today. I pay my respects to the elders past, present, and emerging, and the Aboriginal elders of other communities. My name is Alok Pradhan, and I'm a second year CCMP PhD student with Patrick and Denise. And today, for our speaker, we've got Dr. Cindy Zhang. Dr. Cindy Zhang received her PhD in 2021 from Monash University, Australia, for structural studies on the glucagon-like peptide 1 receptor in the lab of Professors Patrick Sexton and Denise Wooten. She was then recruited as a postdoctoral fellow to the same lab to work on projects aligned to the Australian Research Council Center for Cryo-EM of Membrane Proteins. Cindy was recently awarded an ARC Discovery Early Career Research Award, or DECRA Fellowship. Her research program incorporates cell biology, biochemistry, structural biology, in particularly cryo-electron microscopy, molecular biology, and pharmacology to gain molecular insights into G-protein coupled receptors. Her particular interest is in the structure and dynamics of class B GPCRs to understand how diverse ligands bind to and activate these receptors, which are also involved in many physiological processes and are major therapeutic targets for a range of diseases. And today the topic of presentation will be the structural insights into the activation and modulation of class B1 GPCRs by small molecule ligands. Cindy, the screen is all yours.
Thank you very much for Alok for the kind introduction, and thanks CCMP committee for this great opportunity to share my research that focus on the G-protein coupled receptors, GPCRs. So firstly, a brief introduction about the superfamily of proteins that consisting of 7 transmembrane helices are linked with loops from extracellular side, ECLs, and intracellular side, ICLs. And there are around 800 members in human. GPCRs can respond to a broad range of ligands, including classic neurotransmitters, hormones, photons of light, and are involved in both normal physiological response and diseases. So they are important drug targets for about 35% of marketed drugs. As their name suggested, these receptors coupled to the heterotrimer these proteins coupled to the heterotrimeric G proteins consisting of alpha, beta, and GABA subunits. When the ligand bound to the receptor and G proteins coupled to the receptor and to promote the exchange of GDP to GTP and to activate the receptors, the activated G proteins then dissociate from the receptor, leading to different signaling pathways. And so for the past 15 years, crystallography has been successfully used to solve the GPCR structures in their inactive state. And some of the structures have been used for the structural-based drug discovery to find antagonists to block the signaling pathways. However, for many of the drug targets, the G-protein bound active state is more relevant for the drug development and more important in understanding how the ligands can activate this receptor and leading to different signaling pathways, which is a key focus in our lab. So GPCRs can be divided into 5 major classes. And prior to 2017, there is only one G-protein coupled GPCR structures, that is a B2 receptor from class A, has been solved by using the crystallography. However, this method has few success in other GPCR structures. So our group and others then turned to cryo-EM. And in 2017, our group solved one of the first GPCR coupled G-protein complex structures, a calcitonin receptor from class B1. So now cryo-EM has been widely adopted in GPCR field, and there are diverse receptor structures from all 5 major classes. And my research focused on class B1 GPCRs that can be activated by hormone peptides. So this class receptors have complicated signalings. They can couple to different subtypes of G protein heterotrimer, predominantly couples to GS, leading to the increased level of cyclic EMP, and GOI to block this pathway. They also can couple to GQ11 to promote the intracellular calcium mobilization, and the G12 and 13 to activate different kinases. They also can recruit beta-arrestins, leading to arrestin-dependent signaling pathways. And the receptor also undergoes to the internalization and cycles back to the plasma membrane. So the ligand that act on this class of receptors may also have very complicated behaviors, ranging from full agonist, partial agonist, neutral antagonist, and inverse agonist, depending on which pathway is assessed. So this gives rise to the biased agonism, which describes that two ligands acting on the same receptor may have the potential to lead to the different signaling pathways and leading to the distinct physiological responses. So these class receptors are involved in the physiological responses and are well-established drug targets for various diseases, including bone disease, diabetes, obesity, neurodegeneration, cardiac diseases, and anxiety. So the receptor that I'm going to focus today is an important class B1 GPCR called the glucagon-like peptide 1 GLP-1 receptor. And it is best characterized in pancreas, where it regulates the insulin and glucagon secretion. And these hormones target at these tissues to regulate the plasma glucose level. So it is a well-established drug target for the treatment of diabetes. And this receptor is also widely expressed in the human body with critical functions, including neuroprotection, cardiac function, gastric emptying decrease effect. So it has a potential for the treatment of other diseases. There are around 10 therapeutics targeting at this receptor for the treatment of type 2 diabetes, and all of them are peptide drugs. And we recently solved the structures of this gold standard semaglutide bound GLP-1 receptor and a G protein heterotrimer. And we use nanobody 35 to stabilize the whole complex. So you can see the peptide binds in a 2-domain binding mode, with the C-terminus binding to the extracellular domain of the receptor ECD, and N-terminus insert deep within the transmembrane domain TMD. And despite the high activity of the peptide agonists, they require injections and have considerable side effects. So there is a huge interest in developing orally available small molecules that can mimic the endogenous peptide GLP-1. However, there is a few success in developing small molecules, probably due to that mimicking the bulky peptide binding side of small molecules is extremely difficult. So today I'm gonna talk some progress on the non-peptidic ligands. The first part is agonist, the second part is allosteric ligands. So let's go to the first part. There are 3 GLP-1 clinical non-peptide agonists that have entered into clinical trials. The TT compound has already stopped, but its analogs, including TT-OAD2, is still under investigation. And the recent 2 success with Pfizer in the middle and the Chugai compound at the bottom have entered into phase 2 clinical trials. And in this study, we first tested 6 different signaling and regulatory pathways of both compounds, with Pfizer compound in green and Chugai compound in blue. From cyclic EMP accumulation pathway, we can see both compounds are full agonists relative to the GLP-1, shown in orange. And only Pfizer compound can activate all the other signaling pathways despite a lower potency than GLP-1, including the calcium mobilization, phosphorylation of ERK, recruitment of beta-arrestins, and internalization. And actually, given the very distinct chemical properties of Pfizer compound, it actually has a very similar signaling profiles relative to GLP-1, despite the small differences in beta-arrestin recruitment. In contrast, the Chugai compound in blue was inactive in all the other signaling pathways. So it is a heavily biased agonist towards cyclic AMP signaling. And for TT compound, it also has little activity in other signaling pathway and a bias towards cyclic AMP signaling, but with a partial agonism relative to the other two small molecules. So here give us a question that how does the distinct Pfizer but not Chugai or TT compound can mimic the actions of GLP-1? So in order to answer this question, we solved the complex structures of GLP-1 receptor and the G-protein heterotrimer when bound GLP-1, Pfizer compound, Chugai compound, or TT compound at a global resolution of 2.1 to 3 angstroms. So we modeled the receptor into the cryo-EM density, and we can see their potential differences. Relative to the peptide that binds to the allosteric binding pocket, extending to the receptor ECD and insert deep within the TMD, the three small molecules binds higher up within the receptor TMD. And there are also potential differences on the receptor conformation, including the receptor ECD. So relative to peptide-bound receptor ECD, the ECD when bound with Pfizer and the Chugai compound has a more closed conformation, with the N-terminus covers the binding pocket, while the ECD of TT compound was not well resolved. And an overlap from this, we can see it is actually has a more closed conformation. And there are also potential differences at the receptor TMD from extracellular side. So an overlay of them, we can see there are potential differences on the top of TM1, TM2, and ECL1, but the most pronounced differences is at the location of ECL3 and TM7. When Pfizer compound bound, this region shown in green has a closed conformation. And so this compound has a closed binding pocket, while the Chugai compound induced a larger outward movement of this region shown in blue, and even more than the peptide-bound receptor shown in orange. So Chugai compound has a much larger, and Chugai and TT has a much larger binding pocket, larger than the bulky peptide binding sites. We then take a closer look at the cut-through of the binding pocket with peptide, Pfizer, and Chugai compound with a higher resolution. So I'm going to play this video. We can see peptide has a large binding pocket extending to ECD and deep within the TMD, while Pfizer compound has a completely closed binding site. And the Chugai compound has a much more open binding pocket. And this differences is due to the different location of ECL3 and TM7, as we discussed before. So let's have a closer look at the details of the ligand interactions within the binding pocket. So the peptide has extensive interactions with both hydrogen bound and hydrophobic interactions with the receptor ECD and receptor TMD. And there are structural waters that are well supported from by the cryo-EM density. So the structural waters forms a continuous water network that link the bottom of the peptide to the central polar network at the bottom of the binding pocket. And both N-terminus of the peptide and the central polar network at the bottom of the binding pocket are important for the receptor activation from the structural function studies. And Pfizer compound, with one Pfizer compound in purple bond structure, we can see it binds higher up within the receptor. And there are structural waters that are also well supported from cryo-EM map that link the compound all the way down to the bottom of the binding pocket. And an overlap between the Pfizer compound in purple and the peptide in green. We can see Pfizer compound sits in the vertical orientation overlapping with the middle range of the peptide, and the structural waters in purple overlap with the N-terminus of the peptide. So this explains how Pfizer compound can mimic the actions of GLP-1. While in contrast, Chugai compound, shown in yellow, has a more open binding pocket, and the splayed TM7 exposed the binding pocket to the bulky waters from the solution, so unable to stabilize a continuous water network. And an overlay between two ligands, between Chugai and GLP-1, we can see Chugai sits in a more planar pose, with limited overlap with the GLP-1, so explaining why Chugai has different pharmacological profiles. And combined with different ligand binding mode and receptor conformations, we gain some structural insights into the biased agonism. So now we understand that the ability to interact with the receptor TM7 and ECL3 is important to maintain the signaling pathways other than cyclic AMP. So GLP-1 has this aspartic acid 15 forming salt bridge with arginine on TM7, which in turn forms a continuous halogen network with residues on ECL3 and TM5. And this carboxyl group on Pfizer compound in purple also play a similar role to facilitate the formation of this hydrogen bond network. And there are also, for both ligands, there are structural waters that link the ligand to the bottom of the binding pocket and the receptor ECL3. Well, in contrast, for Chugai and TT compound, they are unable to form interactions with ECL3 and TM7, leading to poor activities in other signaling pathways. So as we noticed from previous slides, there are also differences in the receptor ECD induced by different agonists. So then we further analyzed the conformational dynamics by using 3D variance analysis. So here is the complex maps. Within the middle is a detergent micelle and the receptor ECD extending out at the top and the G protein at the bottom. So I'm gonna play this video and let's focus on the receptor ECD. So relative to the GLP-1 with limited motion, the Pfizer compound and Chugai compound can stabilize the receptor ECD, while TT compound has a large motion of this region, spanning around 20 angstroms. So we also did the ECD truncation experiment. If you still remember from wild-type receptor, the GLP-1 in green, Pfizer in purple, and Chugai in blue are full agonists in cyclic AMP signaling, while TT compound is a partial agonist in this signaling pathway. And the truncation of receptor ECD has no, the truncation of receptor ECD showing in open square has no effect on TT compound partial agonism, but completely abolished the other activities for the other ligands. So these results indicate that Pfizer and Chugai compound can stabilize the receptor ECD in and more mimic the actions of GLP-1. So, and this may contribute to their high activity in this signaling pathway. And the reason for the stabilizing of both small molecules is due to the extensive interactions of both small molecules with this tryptophan 33 on receptor ECD. And this tryptophan is only conserved from human and primate receptors, but not the other mammalian species where it is a serine. So with the when doing experiment with the cells expressing wild-type human GLP-1 receptor show in green, we can see both compounds are full agonists in cyclic AMP signaling. But expressing the mouse GLP-1 receptor shown in orange, it has a little activity. And we also do the mutant. When tryptophan 33 mutated to serine from human receptor shown in light green here, both compounds has little activity on the signaling pathway, while serine 33 to tryptophan from mouse receptor, shown in light orange, the activity is recovered. So this indicates the important role of this tryptophan 33 in maintaining the activation of both compounds. So this has important implications for the preclinical drug development, as they are always tested from the rodent models for the type 2 diabetes and obesity. So the summary of part 1. Firstly, the structural differences involving extensive water-mediated hydrogen bond networks could explain why Pfizer compound, but not Chugai or TT compound, can closely mimic the actions of GLP-1. Secondly, the differences in dynamics also contribute to the agonist-dependent receptor activation. Thirdly, the tryptophan 33 is important in species selectivity of Pfizer and Chugai compound. Then let's move to the part 2 of the allosteric ligands, and specifically into 2 parts, the allosteric agonism and allosteric modulation of native ligands. So another therapeutic strategy is using allosteric ligands that binds topographically different from the orthosteric binding pocket and act in a cooperative manner to modulate the affinity or efficacy or both of the orthosteric ligands, as denoted by the cooperativity factor alpha beta, to define it whether it is a negative allosteric modulator NAM or a positive allosteric modulator PAM. If a PAM also acts as an agonist, we call it agopams. So the allosteric ligands may offer better receptor subtype selectivity, spatial temporal control, and drug safety. And there are several allosteric ligands that have been discovered, and we are interested in 3 of them, compound 2, BTP, and compound 19, which displays a different modulation effect on endogenous peptide, including 2 full agonists, the GLP-1 and oxyntomodulin. However, GLP-1 full length is quickly cleaved by protease, such as DPP4, to generate its main metabolite GLP-1 936, with the first 2 residues, with the lack of the first 2 residues. So this metabolite can circulate in the human body at a much higher concentration than the full length of GLP-1. So firstly, we tested the cyclic AMP accumulation on a concentration-dependent manner of different endogenous peptide. So oxyntomodulin in orange is a full agonist relative to GLP-1 full length in black, despite a lower potency, while GLP-1 936 has a very weak activity in this signaling pathway. So in order to understand how different peptides can engage with the GLP-1 receptor, we also solved the structures with oxyntomodulin and GLP-1 936 bond structures complex with the G protein at a global resolution of 2.2 and 3 angstroms respectively. We can see the cryo-EM densities well supported the peptide binding pocket for oxyntomodulin, and a comparison between oxyntomodulin and the GLP-1 full length shown on the right. You can see they have a very similar binding mode, and even the side chain rotamers, despite they have different sequence, and also they can induce a very similar receptor conformations from both extracellular side and intercellular side. And then when GLP-1 936 bound, we can see the cryo-EM density for the peptide is not very well resolved, so only the backbone of the peptide can be modeled. And and we overlay the GLP-1 936 to the full length of GLP-1 in green, and we can see there is a large movement between them. So the GLP-1 936 moves towards the interface between TM1 and TM2, and also induce potential differences in the receptor ECD. You can see the N-terminus of the ECD moves 9 angstroms towards the receptor TMD, overlap with the full-length GLP-1 binding site. And also there is potential differences on on the receptor TMD, including top of TM1, TM6, TM7, and ECL3. However, the central port network that is important for the receptor activation still remains the active state relative to the ground state shown in black, as well as they can induce a very similar intracellular conformation of the receptor. So to further understand how different peptide ligands can induce different activity of cyclic AMP signaling pathways, we collaborate with Giuseppe and Chris from Coventry University to analyze the conformational dynamics by using molecular dynamic simulations. So we modeled all the regions that were not well resolved from cryo-EM based on the limited cryo-EM density and chemistry, and then we ran the MD simulation in the POPC lipid environment across a microseconds in triplicate. So you can see. On the left, it is GLP-1 bound receptor, and we can see consistent with the static structure, GLP-1 forms extensive and stable interactions with receptor ECD and TMD, while the oxyntomodulin is more mobile, particularly the receptor C terminus, and there is a kink and this middle range of the peptide, as well as the peptide N terminus. Well, for the GLP-1 936, it's just very mobile. It's very mobile within a much larger binding pocket, especially the peptide N terminus that's randomly moving around, which is important for the activation of the receptor. So from these results, we can see the conformational dynamics may contribute to the peptide pharmacology. And then we tested the cyclic AMP signaling on a concentration dependent manner of compound 2 in blue, BTP in green, and compound 19 in red. So both compound 2 and BTP can increase the cyclic AMP level, but compound 2 is a more robust partial agonist, while compound 19 was inactive in the signaling pathway. So we use compound 2 to further study the allosteric agonism by solving the structures bound with this compound to the receptor and complex with G protein heterotrimer at a global resolution of 2.2 angstroms. So here highlights the compound 2 binding site at the intracellular end of TM6, and we have a closer look at this. It forms the covalent bond with cysteine 347 at the end of TM6 and hydrophobic interactions with the residues around. And an overlay between the compound 2 bound receptor in purple and the GLP-1 full-length bound in orange, we can see there is potential differences at the extracellular side of the receptor. However, from the intercellular side, they have very similar conformations, including an outward movement of end of TM6 relative to the ground state shown in black, as well as a sharp kink in the center of this helices, which is a hallmark of all class B1 GPCR activation. So we further analyze the dynamics by using 3D variance analysis incorporated in CryoSpark. We can see relative to the limited mobilization, the extracellular domain is randomly mobile. It's very mobile when compound 2 bound. And this might be due to the lack of allosteric ligands that can stabilize this region. So in order to further understand how compound 2 can activate the receptor, we also run the MD simulations with or without compound 2 presence from the same start active model. So with the compound 2 bound, we can see the end of TM6 maintains in an active conformation, while in the absence of compound 2, the 16347 slowly turns back to the ground state. And the end of TM6, we can make it further, and the end of TM6 rotate back to form hydrophobic interactions with valines and leucine on TM5 to a ground state. So these results indicate that compound 2 can stabilize the active conformation. And then we tested cyclic AMP signaling in a concentration-dependent manner of different native peptides at the presence of different concentrations of PAMs. First slide is compound 2. And we can see compound 2 can modulate all 3 native peptides, but with different effect. Little effect with GLP-1 full length, but more like higher effect with oxyntomodulin and GLP-1 936 to a certain extent. And BTP has a similar role to compound 2, but generally with less effect than compound 2. While in contrast, compound 19 has no effect on GLP-1 and oxyntomodulin, 2 full agonists, but greatly improves the efficacy of GLP-1 936 with alpha and beta over 14,000 to actually the full agonism. So we are very interested in how these different PAMs can differently modulate the 3 endogenous peptides. So we solved the structures of GLP-1 936 bound BTP, compound 2 or compound 19, and oxyntomodulin bound with BTP or compound 2 at a global resolution of 2.0 to 2.5 angstroms. So for the sake of time, I'm only going to talk about the GLP-1 936 bound group. So this is an overlay of GLP-1 936 bound receptors with or without compound 2 or BTP. So here we can see the BTP shown in green has a similar allosteric binding site. And a closer look at this binding pocket, this BTP also forms covalent bond with 16347 at the end of TM6 and hydrophobic interactions with residues around. And with or without these PAMs, the peptide binding didn't change at all, and as well as the receptor conformations, even the side chain rotamers within the binding pocket stays the same. In contrast, compound 19 binds to an alternative site in the interface between top of TM1 and TM2, which is well supported by the cryo-EM density. So closer look at this binding pocket, we can see compound 19 forms direct interactions with the peptide from the sequence of residue 16 to residue 27, which has different sequence between GLP-1 and oxyntomodulin, which explains that the selectivity on the GLP-1 936. Well, this novel binding pocket has been discovered by Lilly, which solved another structures with this PAM bound to a similar allosteric binding site. However, this is also a PAM of GLP-1 936, but this structure is solved with the GLP-1 full-length bond, so with limited structural insights. And in our structure, the GLP-1 936 in the presence of compound 19 shown in purple has a similar peptide binding mode to the full length of GLP-1 shown in green. So it completely changed the binding mode of GLP-1 936 without the PAMs. And although there is potential differences at the receptor top of TM7 and ECL3. So this explains how compound 19 can modulate the GLP-1 936 to almost to a full agonism. Combined with the different ligand binding mode and receptor conformations, we gained structural insights into full or partial agonism of cyclic AMP signaling pathways. So with GLP-1, oxyntomodulin, and GLP-1 936 in the presence of compound 19, we can see this phenylalanine is in a position that forms pi-pi interaction with this tyrosine on TM1, which in turn forms a hydrogen bond network with residues on TM2. And this hydrogen bond network is important to maintain the high activity in this signaling pathway. So they can achieve full agonism. And Chugai compound rotate this phenylalanine on TM7 to a similar position to facilitate a similar polar network. So it is also a full agonist. And the Chugai compound has this fluorophenyl group, also play a similar role. Well, for GLP-1 936 without any PAM bond or bound with compound 2 or BTP, or compound 2 alone bound receptor or TT compound bound receptor, we can see there is no interaction with this tyrosine. So it's pointing down and no hydrogen bond network was formed. And we also run the MD simulations to further analyze how different PAMs, and we take here compound 2 as an example to promote a stable interaction with GLP-1 936. So this is GLP-1 936, as we discussed above, that it is highly dynamic within a much larger binding pocket, and with the compound 2 presence is the whole peptides is more stable within this binding pocket, and with compound 19 bound, especially this N-terminus with less movement. So these results indicate that these PAMs can promote a more stable interactions of this peptide within the peptide binding pocket. So a summary of part 2, the compound 2 and BTP binds to the end of TM6 and can modulate the activation of all 3 endogenous peptides. And secondly, compound 19 binds to a distinct allosteric binding pocket between the interface of TM1 and TM2 and forms direct interactions with GLP-1 936, explaining its selectivity. And thirdly, the differences between static structures provide structural insights into full or partial agonism of cyclic AMP signaling. And MD simulations indicate the binding of compound 2 blocks the TM6 deactivation of the receptor, and compound 2 and compound 19 can stabilize the interactions between the GLP-1 936 and the GLP-1 receptor. So last but not the least, acknowledgement. Much appreciated for our lab leaders, Patrick and Denise, and the lab members, Matt for the data collection and the data processing, Sam, Alva for the excellent pharmacology data, and Harry from Cryo-EM Center of Monash University, Rado from University of Tokyo for the cryo-EM imaging, and our collaborators, Giuseppe and Chris from Coventry University for the MD simulations. And thank you very much.